Hello everyone, my name is Ali Altaqui and I'm a postdoctoral research scholar in the Optical Sensing Lab at North Carolina State University. I joined the SweetAPPS team in August of last year, and the primary goal of the SweetAPPS team project is to develop integrated data analytics together with optical sensing technologies in order to minimize waste and maximize value in production of sweet potatoes. As you can see, the sweet potato app project consists of 4 strategic goals, and I'm mainly contributing to the second goal, namely the strategic goal 2, sensor development, which is supervised by Dr. Michael Kodinov. Having said that, I'm also collaborating with other members from the different subgroups to help in fulfilling our main goal. So the goal of my project 2 is to investigate and develop different light sensing technologies to assist biologists and farmers in identifying different features of sweet potatoes, including its chemical properties, as well as in distinguishing healthy from rotten or damaged sweet potatoes. So, although there are some existing biological techniques that can serve the same purpose, they are often inefficient, time-consuming, and expensive. For example, one biological technique is known as the Polarimized chain reaction, where it requires biologists to take a small sample of sweet potatoes DNA and then amplify it by producing millions of copies of the same sample, and finally examining this sample in details in a laboratory setup. As you can see, this is obviously time-consuming and require expensive equipment. There are also some other types of technique that require performing destructive measurement of sweet potatoes samples, which are obviously completely not practical. So we hope that by developing these different optical sensing instruments, that they will provide in situ measurements of the sweet potatoes while they are scanned in the exeter machine without the need to transfer them to a lab or external locations. The goal here is therefore to provide a fast, practical, economic, and non-destructive solution. So just to give you some background here, the properties of sweet potato vary significantly across different cultivars. So there are over 400 different sweet potatoes types, which greatly vary in shape, size, and color. But more importantly, they also vary in chemical compositions and nutritional value. So here we show 4 different examples of sweet potatoes: the so-called Bayou, the Bonita, the Covington, and the Murasaki. By just looking at these pictures, you can clearly see that they differ in their shape as well as in their flesh color. But let's look in details at what chemical contents we are investigating. So it's mainly carbohydrates, water, and keratin levels. Basically here the keratin refers to the orange pigment found in sweet potatoes, and the higher this value, the more orange the sweet potato flesh is. The carbohydrates in sweet potato also consist of a mixture of simple sugars and starch and other complex carbohydrates. So some of these sweet potatoes contents that we will examine with our light sensor instruments include glucose, fructose, sucrose. These are simple sugars, and lactose and maltose, also simple sugars, as well as more complex carbohydrates too. So we aim to develop again a light sensor technology to enable us to classify sweet potatoes based on their chemical contents. We also aim to build another light sensor structure that help us to distinguish healthy from damaged sweet potatoes. So our goal is to design and test these instruments first in the lab. So once we do that and achieve our purpose or reach a desirable results, we then want to design test beds for these instruments in the Horticultural Research Station in Clinton and hopefully get it to work there. So in simple terms, we want to scan the sweet potatoes by detecting the scattered light from them as they move in the conveyor belt. The setup will include a laser light and an annulus, as well as a detector. We illuminate the sweet potato with our laser light, showed here, and the light will penetrate the sweet potato skin and scatter inside. The light that is going through the detector is a mixture of reflected and scattered light. So the annulus is used here to allow only the scattered light to go through the detector while blocking the reflected light. Then we detect the scattered light and extract its spectrum, the spectrum information. From this, we can then map the spectrum peaks to the chemical contents of sweet potatoes that we discussed in the previous slide. And from this information, we can sort and classify the sweet potato based on their chemical contents. So this process is repeated where sweet potatoes with different chemical properties produce different spectrum. Some of, you see, there are some challenges here. And some of the factors that we will consider in our design is speed. So this means how many seconds it takes to collect the information from a single sweet potato. Or in other words, the number of sweet potatoes detected per minute. Another factor is the sweet potato shape. So giving different sweet potato sizes, as you can see here, given different sweet potato sizes, how are we going to adjust or design our setup to ensure that we obtain consistent results regardless of the sweet potato size? Light throughput and sensitivity is another challenge. So we want to see how much light we can collect with our setup, giving different speed settings as well as different sweet potato sizes. Is the light collected sufficient to produce meaningful results? Also, we will consider the accuracy and consistency. This means that for 2 sweet potatoes with the exact chemical characteristics, will our light sensor instrument produces accurate and consistent results? Or will we need extra post-processing steps to improve our data analysis, such as the use of machine learning? So here is an example of the kind of information that we hope to collect with our first setup. So these measurements were collected in our lab from 10 different sweet potato breeds. As you can see, there are some notable peaks which can be assigned to different chemical compositions. And we want to map these chemical structures to carbohydrate contents or keratin level in the sweet potato, as we have discussed previously. So for example, zooming in this peak at 1,500 per centimeter, which is related to the keratin level, we see that this peak appeared in 5 different sweet potatoes out of the 10 we tested, including the Covington, Biobel, Beauregard, and C13, and other. This peak indicates the level of orange pigment in the sweet potato, and we see that all of these SP types have orange flesh. Meanwhile, those sweet potatoes that didn't display this peak, or didn't have this peak, had white flesh. So from this example, we see that this type of sensor will help us in identifying some, like in this case, the creatine level, but it will help us in general in identifying the chemical contents of sweet potatoes. The other type of sensor that we will develop will assist us in distinguishing healthy from damaged sweet potatoes due to, for example, internal necrosis. Again, our sensor will display information that are also related to the light spectrum. From this information, we can identify whether the sweet potato is healthy, if it yields a result similar to the blue spectrum in this curve, or if the sweet potato is damaged, if it produces a result similar to the red spectrum. So these are briefly the functions of the instruments that we want to develop in this project, with the aim that they hopefully provide a reliable and practical solution to farmers with less time, efforts, and costs. Thank you for taking the time to watch this video.